I'm Helen Walden. I'm a professor of structural biology here at the University of Glasgow. The work that we do in our lab is trying to understand how molecular signals regulate biological processes. In particular, the signals that we study are ubiquitin signals, where a small protein is attached to other proteins in order to change their fate. Ubiquitin signaling is actually a huge signaling system in human cells. When it was first identified, it was thought to be a signal that targeted the proteins it had been stuck onto for clearance, but we now understand that those signals are used for all sorts of different biological processes.
All the proteins that are part of the cells, they're all interfacing with each other. They're all connected. They need to specifically recognize each other, do chemical reactions. These underlie everything. In particular, the one we happen to be most interested in is DNA damage. Throughout day-to-day life, your DNA is damaged. What cells have is pathways to repair this damage. So signaling sort of networks that will recognize the damage and bring in the right machinery. I like to think of it as machinery, but should say in quotations. So that comes in, cuts out the damage, repairs it. And so, yeah, that's molecular signaling.
If you get errors in your DNA and your DNA is not replicated faithfully, this can introduce a mutation. That mutation can change then the functional readout of the protein that gets made from that mutation. Most cancers, for example, melanoma, let's take that as an example. When there is exogenous damage from UV light sources, the DNA that is being replicated in that region may accumulate a mutation that enables that cell to grow quicker than other cells, for example. And that can lead to a tumorigenic phenotype. When I started my lab, I started it at one of Cancer Research UK's institutes. This is the third place that I have established the lab, and I would say this has been my favourite place to work. The collegiality of the environment and just that diversity of science and ways of thinking, I think it's a fantastic place to work. There's a whole burgeoning pharmaceutical field at the moment that exploits the fact that there is machinery inside cells that will stick signals on proteins and direct those proteins for clearance. There's a huge field devoted to trying to hijack this machinery to develop therapeutics for previously, untargetable proteins. I have now a couple of different consultancies working with companies who are trying to develop their own version of these molecules. And there is a lot of activity, certainly in the cancer therapeutics field, targeting that particular enzyme. It's called USP1. And so while we are not doing the drug development ourselves, certainly everybody is now using our protocols for producing the proteins, our protocols for producing, our assays for understanding the enzymatic activity. So it does feed into this development. I went to a conference that had a lot of drug development representatives there, and there are now 3 drugs in clinical trials targeting that enzyme. This is certainly, out of all the work I've ever done, this to me looks like the most likely candidate to actually end up being targeted for some therapeutic.